வணக்கம். நம்ம இன்னைக்கு பார்க்க போறது பயோகெமிஸ்ட்ரியில அமினோ புரோட்டீன்ஸை பத்தி பார்க்க போறோம். வீடியோக்குள்ள போறதுக்கு முன்னாடி this video sponsored by Eduquest.
The struggle to find courses that truly speak your language, it's finally over. Welcome to Eduquest, where learning is made just for you. At Eduquest, we bring you a wide range of online courses, from career building programs to skill boosting masterclasses, all in your regional language. Because we believe language should never be a barrier to success. Our courses are tailor-made after understanding what today's learners really need. So why wait? Start learning the smarter, simpler way only with Eduquest. Visit eduquest.courses and explore your next course today. And hey, don't miss out on our exciting offers and promotions.
வாங்க இப்ப வீடியோக்குள்ள போலாம். Amino acids. Introduction to proteins. Proteins mediate nearly every process that take place inside a cell. They are most abundant biological macromolecules in cells. All proteins, regards of organisms, are composed of the same set of 20 amino acids that are incorporated into them during translation. Due to nearly limited variety in sequences of amino acids in proteins, nearly all imaginable functions can be encoded in proteins. Next, functions of amino acids. Amino acids வந்து ஒரு crucial ஒரு லைஃப் அது என்ன கண்டெய்ன்ஸ் வந்து peptides and proteins வந்து இது know or building blocks for all living things. The linear sequence of amino acids figures in the polypeptide chain determines the three-dimensional configurations of a protein, and the structure of a protein determines its functions. Amino acids are important for sustainable the health of the human body. They largely promote the structures of molecules, muscles, human nerve system, healthy functioning, the health of vital organisms, normal cellular structures, production of hormones. Next, functions of amino acids. The breakdown of both diarrhea and tissue proteins aids nitrogen-containing substrates and carbon skeletons. The nitrogen-containing substrates are used in the biosynthesis of purine, pyrimidine, neurotransmitters, hormones, proteins, and non-essential amino acids. The carbon skeleton are used as a fuel source in the citric acid cycle, used for a glycolysis, or used in a fatty acid synthesis. Next, common feature of amino acids. There are 20 standard of amino acids. All amino acids contain a carbon to which a typical 4 different substitute groups are attached. These groups are alpha amino group or A carbonyl group, hydrogen, and the variable R group side chain. The glycerine, the R group, consists of another hydrogen atom in the another 19 amino acids. The alpha carbon in a cell center. The alpha amino acids and alpha carbonyl groups are charged at the neutral pH. Next, configuration of amino acids. There are 2 possible configurations for these 4 substitutes, the D and L, stereomeres with the mirror images of each other enteromeres. The standard amino acids have the L configuration. Amino acids are classified based on the characters of all groups. இப்போ வந்து இங்கே நிறைய அப்சர்வேஷன் ஸ்டாண்ட் அமினோ ஆசிட்ஸ் தெரியும், அதை பார்த்தீங்கன்னா உங்களுக்கு புரியும். நெக்ஸ்ட், நம்பரிங் ஆஃப் கார்பன்ஸ் இன் அமினோ ஆசிட்ஸ். எப்படின்னா ஒரு கார்பன் ஒரு ஆட்டம் அமினோ ஆசிட்ஸில் அதை வந்து லைசின் ஆல்ஃபா கார்பனை வந்து ஆல்வேஸ் கார்பன் 2 அமினோ ஆசிட்ஸில் பண்ணுவாங்க. ஆல்ஃபா கார்பனைல் குரூப் தான் என்ன இங்கே பண்ணுவாங்கன்னா கார்பன் 1 பண்ணுவாங்க. கிளாசிஃபிகேஷன் ஆஃப் அமினோ ஆசிட்ஸ் by Hall group. Amino acids are collected into different categories based on similarities in the properties of their R groups. One such classification scheme relies heavily on the polarity of R groups. The amino acids are classified into 5 categories based on the properties of R groups: non-polar and aliphatic, aromatic, polar and unchanged, positively charged, and negatively charged. First type is non-polar aliphatic amino acids. The amino acids in this group lack of polar functional groups in their side chains due to the hydrophobicity of the R groups. They are often clustered together within the interior of protein. Stabilization protein structure via hydrophobic instructions. இங்கே நான் வந்து செயின்ஸை போடுறேன், அதை பாருங்கள். Next, aromatic amino acids. The side chain of the aromatic amino acids purine, tyrosine, and tryptophan overall are very hydrophobic. The H group of tyrosine also contains a polar hydroxyl group that can participate in H bonding interactions. The H groups of tyrosine are particularly tryptophan absorb ultraviolet light at the maximum of 280 nm wavelength. Next, polar uncharged amino acids. The H group of the polar unchanged amino acids all contain polar functional groups that can hydrogen bond in with water. Serine and threonine control hydrolyl groups. Asparagine and glutamine contain aminate group, and cysteine contains a sulforyl group. Those polarity is quite weak. Alpazine and glutamine are the aminate forms of the two negatively charged amino acids, asparagine and glutaminate. Next, serrate and diphosphorate bonds. The triophyl groups of two serrate residues are readily oxidized to form a covalent link diametric amino acids known as cysteine. In cysteine, the two serrates are joined by a diphosphorate bond. The diphosphorate link cysteine reduces strongly hydrophobic. In proteins, disulfide bonds form covalent links between the different parts of the polypeptide chain or between two different polypeptide chains. Positively charged amino acids. The most hydrophobic groups are those that are either positively or negatively charged. The child chain of lysine and asparagine are fully positively charged at neutral pH. In lysine, a primary amino group is attached to the alpha carbon of the side chain. In azarine and glutarine group of the side chain is positively charged. The histide R group contains an aromatic indrosyl group that is positively charged at neutral pH. Histine reduces the function in many enzymes, catalyze the reactions as protein donors and other acceptors. Next, negatively charged amino acids. The H group of the asparagine and glutamate contain carboxylase group that are fully negatively charged at neutral pH. In asparagine, carboxylase group is attached to the beta carbon of amino acid backbone. Glutaminate, the carboxyl group is attached to the Y carbon. Okay, thank you.